welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here's what I bring to you all from the world of medicine. The difference in development of stem cells between modern humans and Neanderthals. To investigate the significance of these 6 changes for neocortex development, the scientists first introduced the modern human variants in mice. Mice are identical to Neanderthals at those 6 amino acid positions, so these changes made them a model for the developing modern human brain. Felipe Moura Bermudez, the lead author of the study, describes the discovery as: we found that 3 modern human amino acids in 2 of the proteins cause a longer metaphase, a phase where chromosomes are prepared for cell division, and this results in fewer errors when the chromosomes are distributed to the daughter cells of the neural stem cells, just like in modern humans. To check if the Neanderthal set of amino acids have the opposite effect, the researchers then introduced the ancestral amino acids in human brain organoids, miniature organ-like structures that can be grown from human stem cells in cell culture dishes in the lab and that mimic aspects of early human brain development. In this case, metaphase became shorter and we found more chromosome distribution errors. According to Moura Bermudez, this shows that these 3 modern human amino acid changes in the proteins KIF18A and KNL1 are responsible for the fewer chromosomes distribution mistakes seen in modern humans as compared to Neanderthal models and chimpanzees. He adds that having mistakes in the number of chromosomes is usually not a good idea for cells, as can be seen in disorders like trisomies and cancer. Our study implies that some aspects of modern human brain evolution and function may be independent of brain size, since Neanderthals and modern humans have similar sized brains. The findings also suggest that brain function in Neanderthals may have been more affected by chromosome errors than that of modern humans, summarizes Wieland Huttner, who co-supervised the study. The gut patrol. A new study in Science Immunology reveals how the barrier cells that line the intestine send messages to the patrolling T cells that reside there. These cells communicate by expressing a protein called HVEM, which prompts T cells to survive longer and move more to stop potential infections. Barrier cells or epithelial cells form a 1-cell thick layer that lines the gut. One can picture these cells lining up like a busy queue outside a nightclub. The epithelial cells squish together. They jostle each other and chat. Meanwhile, T-cell security guards circulate around the line, looking up and down the block for signs of trouble. These T-cells move around the epithelial cells as if they are truly patrolling, says Cronenberg. But what keeps these T-cells in the epithelium to do their job? We have got some insight on what gets T-cells to the gut, but we need to understand what keeps them there, says Cronenberg. In fact, a lot of immune cells reside long-term in specific tissues. By understanding the signals that keep T cells in certain tissues, Cronenberg hopes to shed light on conditions like inflammatory bowel disease, where far too many inflammatory T cells gather in the bowel. In the new study, the researchers found that important signals in the gut are sent through the basement membrane, a thin layer of proteins beneath the epithelium. In our nightclub scene, the basement membrane would be the sidewalk where everybody stands. Their experiments show that epithelial cells receive signals through HVEM proteins on their surface that stimulate synthesis of basement membrane proteins. The team found that without HVEM, the epithelial cells couldn't do their job because they produce less collagen and other structural components needed to maintain a healthy basement membrane. T cells detect the basement membrane via adhesion molecules that they express on their surface called integrins. The interaction of the T cell integrins with the basement membrane proteins promotes messages that allow the T cells to survive and patrol in the epithelium. It is as if the epithelial cells have written messages on the sidewalk: stay here, patrol here, do your job. Without a sufficient basement membrane, T cells would not survive as well or go on patrol. Using a mouse model, the researchers then showed that removing HVEM expression only in the gut epithelial cells was a major blow to gut health. Patrolling T cells could not survive as well and they didn't move as much. These T cells made lousy security guards. When challenged with Salmonella typhimurium, an invasive bacterium that causes gastroenteritis, the T cells allowed the infection to take over the intestines and spread to the liver and spleen. Therefore, HVEM from epithelial cells laid the groundwork for T cells to guard the gut. It was the very reason they survived in the epithelium, communicating with the T cells indirectly through the basement membrane. new research published in JASN which reveals sex differences in the recognition, monitoring and treatment of chronic kidney disease. A team led by Juan Jesus Carrero studied a variety of CKD care indicators among 2,27,847 people with a first ever detected low level of kidney function, denoting probable CKD in Stockholm's health system from 2009 to 2017. The investigators found that compared with men with similar characteristics, women were less likely to receive a diagnostic code related to CKD, be referred to a nephrologist, and have their kidney function monitored. Additionally, women were less likely to receive guideline-recommended medications. This study identifies healthcare gaps that may explain previously reported sex differences in the prevalence, progression rates, and outcomes of persons with chronic kidney disease, said Dr. Carrero. Analysis of time trends during the last decade showed that many indicators are improving over time. For example, there has been an increase in the rate of certain tests of kidney function over the years, but the rate of testing among women with CKD has been persistently lower than that of men. We are unable to identify the reasons for this potential undermanagement and speculate on possible causes such as challenges in interpreting serum creatinine, a marker of kidney function and a waste product of the normal wear and tear on muscles of the body, in women who on average are smaller and have lower muscle mass than men, one of the researchers noted. It's also possible that subconscious biases operate among healthcare professionals, believing that CKD is less problematic in women or that women themselves are more likely to deny the disease. In any case, our study brings attention to healthcare gaps amenable to correction, the authors noted. The impact of sleep deprivation on kids on cognitive development. The findings were published recently in the journal Lancet Child and Adolescent Health. The American Academy of Sleep Medicine recommends that children aged 6 to 12 years of age sleep 9 to 12 hours per night on a regular basis to promote optimal health. To conduct the study, the researchers examined data that were collected from more than 8,300 children aged 9 to 10 years who were enrolled in the Adolescent Brain Cognitive Development or ABCD study. They examined MRI images, medical records, and surveys completed by the participants and their parents at the time of enrollment and at a 2-year follow-up visit at 11 to 12 years of age. We found that children who had insufficient sleep, less than 9 hours per night at the beginning of the study, had less grey matter or smaller volume in certain areas of the brain responsible for attention, memory, and inhibition control compared to those with healthy sleep habits, said corresponding author Zhe Wang, who is a PhD professor of diagnostic radiology and nuclear medicine at UMSOM. These differences persisted after 2 years, a concerning finding that suggests long-term harm for those who do not get enough sleep. In follow-up assessments, the research team found that participants in the sufficient sleep group tended to gradually sleep less over 2 years, which is normal as children move into their teen years, where sleep patterns of participants in the insufficient sleep group did not change much. The researchers controlled for socioeconomic status, gender, puberty status, and other factors that could impact how much a child sleeps and affect brain and cognition. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.